Okay, everybody, welcome back to our second session, which will be named Modeling Microbiota-Wide Metabolism with Mycom. And as we motivated it, we will now gonna take a little look into like the functional consequences of the different microbiota we observed in the first half of the course. Also, if you joining just for that second part of the course, that just reiterates that we are also aware that like a lot of people might be in different time zones right now. So all of those course materials, including like the presentation I'm giving right now, will be available as videos. The presentations are available online, the notebooks are available online, and the Slack, the support channel, will also remain open for, I think, at least this week. So if you're, for instance, like if it's 2 o'clock in the morning, like for you right now, and you're basically dying of tiredness, like you can also just continue doing that course tomorrow and you will just receive the same support. You can still do the exercises and ask for help. Will all work out. So don't worry about it. Okay, well, let's get started with that second part of the course. And again, like before, like it might be helpful to actually open up the slides, and I'll ask the TAs maybe to just post that link again. Actually, if you have the previous slides open, it's actually very simple. You can go to the address bar and change the last part, which was 16S, to MICOM now, and it will just open up those slides for you. If you don't, you can just click on the link in the Slack or the one you received in the email or on the course website. It's on all of those. And just a quick reminder, and maybe also for people that have only joined for the second part, we have those 3 parts that will be running. So there will be that presentation that I'll be using to explain things, but we will be actually running the analysis in our Google Colab notebook, which we will open in a second. And we also have that chat. So if you haven't joined the support Slack, please do so. You'll find that link in the chat of that Zoom call, and also in this in the email that was sent to you, and that you can ask any questions there. Those will be relayed to me if something like really bad happens, but you also get like immediate support from our TAs with anything you might find a bit difficult to follow. Okay, so now we will be opening again like a notebook, but now we'll open a notebook for the second part of the course. I'll be using that link here, but you can also use the link that is probably posted in the Slack right now, or the one you received in the email. So, and we were just doing exactly the same thing that we did before. So we're opening up that notebook. It looks pretty similar. Some more explainers here. And again, like the structure is the same. We have those cells that are either text and we have parts that are code. In this step, for most of the course, we'll actually be running Python code. So we won't need that initial exclamation mark, and only for the exclamation mark, only for the first part where we install some things, and later on we actually use pure Python code. And that thing works the same. If you want to connect to the runtime, click the connect button on the upper right here. If you don't see the connect button, it shows you something like RAM and disk. You're already connected, so don't worry about it. And if you're connected, it should look like that. So you see the free RAM and the disk space you have. And again, on the left side, we kind of have the table of contents. On the lower region, we have that folder symbol that shows us all the files in the notebook, which we'll be using. It's the same thing. But yeah, we'll be using it in a second. Right now, let's start with a little bit of introduction, what we actually want to do right now. So I already said that in that second part, we were really interested in function analysis. So we analyzed our microbiome data. So we basically went from raw sequencing data to abundances of microbes. We know which microbes they are. We have the taxonomy. But taxonomy isn't really a perfect explanation of what's really happening in the system, right? So we might be interested, okay, like if there are like particular microbes associated with a disease, why is that so? Like what is the actual mechanism that underlies that? Is there, for instance, a metabolic interaction, which is what we mostly will look at? And there's several approaches to that, which I would say there's basically the most common ones that we've used a lot is basically gene abundances. For that, you basically need to do metagenomics. Everything else doesn't really work that well in practice. Well, somewhat. So for metagenomics, you, like we said in the first course, you actually try to sequence entire genomes of everything that's in our sample. And if you do that, you can simply count the genes that are in there. There's certain analysis pipelines that basically go through that. For bacteria, it's not really hard to find out what a gene is because they always start kind of with a similar type of promoter. So it's really easy to identify open reading frames. So you can do that de novo. You don't even have to know what that gene does. Then often you compare those genes that you found to databases and you can quantify, okay, that particular phosphofructokinase is pretty abundant in that sample, for instance. That gives you genes and gene abundances, and that can be pretty informative, but it has like a small caveat, and that is that gene content is basically only a capacity or potential. So it tells you that the gene is there. But for instance, you might have a gene that helps you to break down a particular starch, but if you don't eat starches, that gene just sits there, right? It's not going to do anything in your gut because it's never really encountered any starch, so there's no effect to it. And that's actually quite common in bacteria. Bacteria are really good in adapting to different environments, so they kind of have a lot of genes to deal with different environments. But if they don't find that environment, those genes will just sit there. They might not actually do something. So that's something we have to keep in mind. Other more elaborate methods, you can use metatranscriptomics. Then you see that genes are actually expressed, which is already a bit better than just having the gene, because the gene might be there, but it might be in an operon that's switched off. So you're actually not even expressing the gene at all. There's no associated protein. You could use proteomics and look at the proteins that are there, which is like even better to see if there's certain enzymes that are actually present, and even do metabolomics where you actually measure metabolites. And if you measure metabolites, you could kind of see if there's particular metabolites that associate with certain microbes, for instance. All of that has a few caveats, and I will kind of start off with a bit of a hot take here. And sorry if anyone feels like offended. It's not really meant really mean, but like just saying that genes and metabolite abundance are really cool, but they might not what you really care about. And why is that? Well, for instance, I could go home and eat a bit of kimchi, which has like a lot of really good bacteria. And those are bacteria you might not actually find in our gut that commonly if you don't eat kimchi. Then afterwards, I eat the bubble gum, and because I'm not really careful, I swallow it. So now you will see that certain kimchi bacteria and the bubble gum are both present in my gut, maybe in high abundances. I would even say they're related to each other. And we conclude from that the kimchi bacteria are producing bubblegum in my gut, which would be a quite interesting paper, but probably not that one that would really be considered like good science. So just because two things co-occur doesn't mean that one, for instance, drives the other, right? So how do we do that? Like, what is the quantity we actually want to see? And for that, we have to look in a little bit how metabolism usually works. So what we might be way more interested in are fluxes. So fluxes is actually a transport of mass across a system. So a flux is a rate of mass conversion, whereas concentrations from metabolites are usually measured, for instance, in millimolar. Flux is actually measured in millimolar per time unit, for instance, millimolar per hour. And for bacteria, we usually also normalize by the gram dry weight of the bacteria that is present in the system. Why is that? Well, because 10 bacteria probably eat way less metabolites than 1 million bacteria, right? So we kind of have to normalize that a little bit. Fluxes are like super informative because as we see on the right, that's actually what's happening in the system. We can actually see the flow of mass through the entire system. So we can, for instance, see that like a lot of glucose gets converted to pyruvate in that example. And that's kind of one of the caveats if you measure metabolite concentrations, for instance, since glucose gets, for instance, absorbed by bacteria so quickly, they love to eat glucose. They're really like sugar freaks. So you actually don't see high concentrations of glucose in your gut usually. Whenever it comes in there, it's either your gut will take it up or the bacteria will eat it up, but the concentrations will be really low. So, and that's maybe a bit counterintuitive, right? It's like one of the most important metabolites for that system, but it's really not there, like, or measurable in that abundance. So that's kind of what we have to take into account. But if we would look at a flux, we would see that there's a massive transport of glucose into bacteria. So that would really tell us, okay, that's something that's important. So fluxes are super interesting, and honestly, that's like not new. We have known that for quite some decades that fluxes are cool, but the problem is that they're really kind of expensive to measure. So one thing I could do, I could longitudinal metabolomics. So if we go back to our bubblegum example, what I could do is I could eat kimchi And then I could kind of track myself, like every hour I kind of measure all the metabolites in my gut and I see if there's actually bubblegum produced. Like I start off with little bubblegum and over time it gets more. Then I could kind of say, oh, those bacteria really produce it. That is actually kind of a flux, right? Because I could say, oh, I produce like, I don't know, 2 bars of bubblegum every hour in my gut. So that would be like more of an evidence that it's really produced. There's also stuff, also methods, not stuff, that could produce fluxes, and you don't even have to measure like longitudinally. And that's basically the trick is you can give, for instance, glucose to bacteria, and then you label the carbon like with your radioactive probe. You make it radioactive, so you can actually probe it. And then if you look after a while, you take a single sample and you can actually just check, okay, I know I gave a glucose where every carbon was kind of radioactive, right? So now I look at pyruvate and see of all the pyruvate in the system, like how much of that pyruvate actually has like a radioactive carbon. And that will kind of tell us like how much was converted to pyruvate. So that's like another trick. And that's even more expensive, unfortunately, than longitudinal metabolomics. So there's definitely a cost factor. 16S sequencing, for instance, can be done really cheaply, but that is really hard. And that was kind of the stage which we started as well. We kind of wanted to get like functional insights, but often what we had was like, well, in good cases, metagenomics, in bad cases, only 16S data. And like I said, for gene content, you do need metagenomics. So that functional analysis wasn't possible. There's some tools like PyCrust, which kind of like what they do is if you know that there's a certain taxon and you have a reference genome, you can kind of predict what the gene content might be, but the taxa in your gut might actually not be exactly the same as the ones that were sequenced, right? Like mostly in the lab, we maintain like lab strains, which are like really easy to grow in the lab, but they might not actually be super informative. So that's something we have to account for here. But yeah, we're still interested in fluxes, we said, right? Because we might only have 16S data or metagenomics. So how do we get there? And that's basically what flux balance analysis does. Flux balance analysis is actually a computational method. And it's an input. It can go from actually genomic data. And the question is, can we infer the most likely fluxes in a biological system? And it has been used quite extensively, but we haven't seen it that much in microbiome science, but it has been used a lot in bioengineering. Like a lot of chemicals these days are produced by bacteria that are kept in really huge tanks. And to kind of design a bacteria that produces a lot of something, you want to maximize the flux, right? So that's where they use flux balance analysis to actually like find particular mutations that they can introduce in the bacteria so it produces the actual chemical that you want. So how does it actually work? Like how can we computationally get like fluxes? And there's a certain assumptions that we make and like certain concepts that we will introduce. So the first one is kind of the flux cone. So the first thing that we often assume is that we have like a constant flow, or what is often called a steady state. And that can be imagined a little bit by if you open your windows right now to like let air into your room, which is especially during a pandemic, a pretty good idea, right? So if you open that window, air will come into your room, but it also obviously air will come out, right? You exchange like old air with fresh air, basically. And while that happens, obviously there is a kind of a balance, because if air would just be coming in and not the same amount of air is getting out, well, the apartment would blow up, right? It would just like fill up with air like a balloon, and at some point it would pop. It could also be the opposite. Air is coming in and more air than is coming in is actually getting out, which would create a vacuum, and then it would implode. So both of those obviously don't happen. Like nobody had his apartment blow up just by opening a window, at least if nothing is in front of the window. And so there is a certain balance that stuff that's coming in is also kind of equilibrated by stuff that's going out. And that actually works for a lot of physical systems, and it kind of also works like that for metabolism. For instance, like in our state, like bacteria grow in our gut, but then we also poop. So it's kind of like an equilibrium with those two processes. And an example, we can actually formalize that. So for instance, we could look at that example biochemical reaction or chemical reaction here, where we have 2 hydrogen molecules like with an oxygen molecule, and kind of converts to 2 water molecules. And if those fluxes are imbalanced, like what I can do is I can basically, everything that's produced in the reaction, I give it like a positive sign and multiply it by the stoichiometry of the reaction. Everything else I give a negative sign because it gets consumed, so we have less of it. And I would say that they're balanced. So the entire balance of it must be 0. So That's what we see in the balance equations. And then we can also put constraints on it. For instance, we can say, okay, in this reaction, I actually want water to be produced. And the reaction should only go in one single direction. So it can only go from hydrogen and oxygen to water, but water cannot like deassemble into oxygen and hydrogen. So and then that would basically mean I say, okay, that particular flux is larger than 0, for instance. That's like maybe not something that happens in bacteria, that particular reaction, mostly because of the huge explosion that it entails. But that particular chemical reaction is a region that why we use helium in balloons and air chips and not hydrogen anymore. So what that actually means is that there's like a limitation, since like I need a particular balance of hydrogen and oxygen to kind of form like water. It's not all possible values of them are possible, right? Like if I have like one hydrogen and one oxygen atom, can't really produce 10 water molecules. So that's kind of a limitation, and that can be formalized like by like a set of linear equations that kind of limit the space of possible fluxes. Like there's only few flux distributions that are actually valid in that setting. And I could put constraints on it, like for individual fluxes, I could say like they can also go in a reverse reaction, then they will become negative. Usually we split up those reversible biochemical reactions, say there's a forward and reverse direction. And so there's one reaction for each. The flux are usually positive. And if I do that, then basically one trivial solution of that equation is that all fluxes are 0. If you multiply anything with 0, obviously it's 0. So the 0 point is definitely included. And then it feathers out into that flux cone, which is the space of all possible fluxes. And flux balance analysis is basically hopefully trying to find that one single point in that space that's actually the most biologically relevant one. So how do we make it more relevant? Well, we can actually look at growth, right? Because if we know, for instance, bacteria are present in our system, well, they can only be present if they can grow. Otherwise, they would be pooped out, for instance, by us, and then they would be gone at some point. So one assumption we can basically make is that we can say, okay, we want to find basically that part of the flux cone that corresponds to production of maximum biomass. And that's fairly easy. Biomass is essentially a flux. It requires a lot of components that have to be coming in to continuously produce biomass and let the bacteria multiply. And if that actually gives like a subspace. So in that flux cone, we have like the red area that corresponds to maximum biomass. And the cool thing about flux balance analysis is that we can incorporate a lot of information. Like we incorporate all the biochemical stoichiometry, and that we can actually get just from a genome. So if you have a reference genome from organism, we can look at all the enzymes. For most of the enzymes, you kind of know which biochemical reactions they catalyze and what's the stoichiometry of that reaction. And by that, we can infer like that entire stoichiometric matrix. And like I said, we make that steady state assumption. And in practice, for bacterial growth, that kind of means we're kind of in the exponential phase, because we're in a phase where the growth rate is constant. If we produce biomass at a constant rate, our growth rate is constant. So we're kind of in that like exponential growth phase of bacteria. And bacteria like to spend a lot of time in that growth phase if they can, but it's an assumption we have to make. And reversibility for reactions, that can usually be determined by thermodynamics. We know that certain reactions can only happen in one direction, others don't. But I also have like fluxes that actually symbolize like import in the system, so things that are coming into the system from the outside. So I can actually incorporate the environment as well. So for instance, I could model that with different diets. So that's like a little bit something that would be really hard with metagenomic data, because you only have gene content. You don't really like think about how the diet might influence that system. So we have that sub area that's maximum biomass, and now like a lot of flux balance analysis about like an art of finding either a range or like even a single point in that space that's like the most representative one. One that actually works really well is finding a parsimonious solution. Parsimonious kind of means find one that's kind of uses the least amount of enzyme. And it basically means I try to keep most of the fluxes as small as possible. And why is that? Because the flux itself is a function of the substrate that's available, how much substrates do you have, but also how much enzyme do you have. And obviously it's costly for bacteria to produce more enzyme that they actually need. So if they can get maximum growth by producing the minimum amount of enzyme, that's something that a lot of bacteria might want. And if you use that it's kind of representative of like kind of walking down the axis, finding kind of like a short path to that particular biomass region. that reproduces experimental fluxes in E. coli very well. So it has been shown that if you use Parsimonious solution, it's actually really good. It's even better than, for instance, incorporating transcriptional data, like to validate that. And it's like kind of based on that assumption that bacteria do not like to produce more enzymes than necessary. So what does Mycom do? Well, Mycom is basically an extension of flux balance analysis to microbial communities. That's where the name comes from. Mycom, microbial community. So it kind of incorporates all the data that you give it. So initially, you usually need to give it at least like the abundance of the bacteria in your system. And that's a bit of a caveat there that like it actually wants it in gram dry weight, but we sequence. So what we have is more like relative abundance of the reads. So DNA extraction has a bit of a bias, and maybe some of the TAs can post Amy Willis's paper on bias there a little bit. She analyzed that really great. So there's kind of like a bias. So the DNA extraction will break down certain bacteria better than others. So you might get like more reads from one bacteria just because of that. But like the orders of magnitude are more or less correct. And you can also specify particular diets that you assume that are present. It will build up basically a huge stoichiometric matrix that does not only contain every single biochemical reaction from each organism in your sample, but also all exchanges between them, and also with like the external environment, which in our case, for instance, would be the gut lumen. It can identify unique growth rates basically for each of the bacteria. And from those growth rates, it also identifies all the fluxes. So it kind of tries to reduce that even bigger flux space into like a smaller region. And then you can use stuff like Parsimonious solutions to kind of go back to like a most likely flux distribution. And if you have that data, you actually get kind of a really, really detailed view in your system. So it is still like a prediction, but it's like an extremely detailed one, which will tell you every single flux and every single bacteria. And those are hundreds of thousands. So it will tell you exactly what is produced by E. coli, what is imported by E. coli, what does E. coli consume inside of itself, and how does it convert it, how much is produced into biomass, and so on. So that's like kind of a cool tool for that. And we basically have two interfaces for that. We have like a QIIME2 and a QIIME2 interface, but also a Python interface. We'll be using the Python interface, but they're almost exactly the same. Honestly, they produce the same visualization. It's the same like algorithm. And yeah, it just like the Python interface is a bit faster to install. So we'll use that just because. And we can do a little bit more with it in certain settings later on. But like most of the workflow you can do with either. So yeah, feel free to use either in your daily work pipeline. Okay, but okay. Now I rambled on way too long. Well, not way too long, a little bit. So let's actually do something, right? So the first thing we will do is run some setups. There's one thing that Mycom actually, so it solves really large numerical optimization problems. And there's actually two different ones it solves, quadratic and linear ones. And it's really hard to find solvers that can do that well. There's obviously a few commercial ones. So unfortunately, those kind of problems also rise a lot in industry. They're very valuable to a lot of companies. So the algorithms or the programs that do so can be solved pretty expensive. So IBM has one and Gurobi also has one. And usually they're very expensive, but they're actually free for academic use. So you can actually just apply if you're a student and you will get it for free. We actually use an open source one that's pretty recent, which is OSQP. It's a great solver for quadratic programming problems. It's as good as the commercial ones always. It's a bit weaker with linear problems, which we also will solve with quite a few of, but it will work for our tutorial. The only thing I have to mention is like, we will lower the accuracy a little bit. So like the numerical accuracy, since all of those are numerical algorithms, like 10 to the minus 3, which kind of means if something is smaller than 10 to the minus 3, we cannot really say if it's really different from 0. So we kind of have to be careful. If you use a commercial solver, that can be lowered. But yeah, it's just kind of a cutoff where we say, like, if a flux is 10 to the minus 4, it might actually not even be happening. So we will start by kind of loading in the material again. Oh, sorry. You might want to do your own copy again. So you can go on File, save a copy and drive again. Rename it if you want. and then we run it. So basically you can start with the first one. If you copy it to your own drive, it will be trusted. You can also run it directly, just nothing will be saved. So that might not be super convenient, but yeah, it's possible. So that will again download all the materials. We'll also download actually the last abundance tables that we had, because that's what we're going to use. We're actually going to use exactly the data that we generated in the first tutorial here to actually connect it with MICOM. For MICOM, usually you can just install MICOM with pip, for instance, if you use Python. It's like super simple. In this case, we do actually do quite some extra steps to get OSQP support. That will be official in MICOM pretty soon as it gets into Cobra Pi. But right now we kind of will install some development versions, so that looks, you actually don't have to like really look through that in that much detail, because that's nothing in the future that won't be necessary anymore. And it's basically just an arrangement that we can run that course with open source software. And that's what all that does. So basically that like pulls in the latest development versions of the OSQP support, so we can actually use that solver here. So we run through all of those. And then in the last step, we also want to enable the QIIME2 interactions. And since these tables are like kind of saved in the biome format, we will install that as well. That will give us access to the biome format and reading those tables that QIIME generates. And that's all we need to do here to install it. Now we have MICOM set up. It will run just fine. We'll take a few seconds here, but yeah, we're done. So that should all work. So you can go through the entire setup session, basically down to the big parts that says MICOM. And just to reiterate, that's what we kind of want to do today. So we will start out with our raw abundance data. We will need a model database. Like I said, like since we map SOT16S data to metabolic models of the entire organism that contains like all of the biochemical reactions, we kind of need that from somewhere, right? So we could map it to existing genomes and then like reconstruct metabolic models, but that might be pretty costly. We actually provide a lot of pre-built databases. The ones we're using today has about 800 strains in there. And then if we have that, the abundance in the database, we can actually build up the entire model, and then we can start simulating growth basically. So we will basically find the fluxes that maximize growth and do some tricks to get that really well and get all the fluxes as results. Okay. To kind of see what is the input data that MICOM actually wants. So how does that look? And to kind of get an idea, we will actually load in some example data that comes with MICOM. It's a really constructed example where we have like 4 different E. coli strains, and we kind of want to see how that looks. And basically what we need actually is, for the abundance table, you kind of combine all the abundance and taxonomy information in a single table. So now, like, you have basically a column where you will say, okay, what's the genus, what's the abundance, and which sample? It's kind of the combination of both and kind of a long data format. The file comma is actually, that's just an example. It's not required if you use a database, which was like before in the initial days of MICOM, it didn't actually use databases. It built up all the models on the fly, which actually is slower. But yeah, but the thing is, okay, that's not exactly what we got from QIIME, right? We got QIIME artifacts, and taxonomy was a separate artifact from the abundance. So that looks like we have to do some work, but actually, we don't. We can actually just do it. Because MICOM actually has an import function for that. And before we actually get there, we will download like the model database, and we will use like the Agora database that comes from Ines Thiele's lab, and Ines will actually give a talk in our symposium tomorrow. So you will be hearing from the very experts on that topic, where they really painstakingly, with a lot of work, reconstructed metabolic models that were actually manually curated. So they reconstructed by now for about 800 bacterial strains, metabolic models, but then actually went and validated them. For instance, they checked, oh, if the metabolic model predicts that you can grow on that growth medium, does that actually work? And then they went back to create the model. So they actually did reproduce like the actual phenotype. And they, for instance, they checked if that organism is known to be able, for instance, to assimilate nitrogen. Is that actually true? Things like that, for instance. So So they actually like defined also metabolic functions that must be compatible. So it's like it's a really well curated database, and Agora is kind of specialized for the gut microbiome. So the strains contained in there are the most common ones that are found in the human gut. So it's perfect for our data, right? So we will download that, and we will download that on the genus level. So those models are already summarized on the genus level because 16S usually gives us like good information down to the genus. Species is usually really hard. So that's kind of the level we will use. So we'll download the database. It's a little bit bigger, still should be fairly quick. And now we have our model database. And now we will also import the data. And that's in the treasure chest folder. That's the same data that you actually generated in the last tutorial. What we will do is basically, we just import the QIIME data. You actually don't need QIIME to install to import QIIME data. So for instance, you can get that from a collaborator that has used QIIME and just use it with MICOM without even installing QIIME. It knows how to manage that. And you can also say what if you want to collapse it. And since our model database will be genus level, and that's kind of the best taxonomy information we get from 16S, we will also collapse on the genus level. What it will basically do is it just takes those tables and builds up that table that we actually need, like the taxonomy table for MICOM. Okay, that will run. You will not really see any output, so don't be too surprised. If you actually run the next cell, it will actually show you what that looks like. And if we look at it, it's exactly what we wanted. So we have a sample ID. We have the abundance, which is just the number of reads of the particular genus in that sample. We have the name of the genus, for instance, Akkermansia here, Bacteroides here, and an ID, which in this case is the same as the genus. You can actually like build up rules to make the IDs more informative. Like sometimes you have like weird names that have spaces and you want to substitute with underscores or something, but yeah, for now it does it automatically. And also gives us a relative abundance. That's kind of the proportion. So for instance, we see that there's already quite some differences. So that sample has like not even 1% of Akkermansia, but that sample has 82% of Akkermansia. So that's a huge difference in here, for instance. And maybe just to comment a little bit on those levels. So if you give data to MICOM, usually there's like several ways how it can later match it to a database. For instance, you see here that it only has a genus column. It could also have columns, for instance, for the phylum, the domain, like for the phylum, the kingdom, the family, the class, and order. The more columns it has, the more it will also use to match it to the database. It will always require that all taxonomic ranks in this table are also exactly the same in the database. That can sometimes be a bit of a problem because different databases use different systems for that. So for instance, Green Genes might call it Prevotella, and then like Agora might call it something different. It doesn't, but sometimes that can happen, even though the species is the same. So there can be less matching to the database if you assume that the entire taxonomy has to line up. So that can be like a small problem, but we will actually only match on the genus name right now, which will work for our data set. That might actually be bad if we have like two different organisms that happen to have the same genus name, but in a different family. That's definitely valid in the taxonomy. It doesn't happen very often, but it can. And then we would mismatch. So it's a good idea to kind of later kind of check that it did it well. But for now, it will work. Okay. What we can also do is we can read in our metadata from before, and we will kind of just merge that into with our taxonomy table. So here we use pandas, which is like a Python package to manage like data frames in general. And we will kind of merge those two on the sample ID. And basically that gives us a bit of an expanded table where we now have the disease status as well, make it a bit easier for us to observe what's happening. Okay, perfect. And the first step we will do, and that will be fairly quick, is we will actually build the community models. It still takes a bit of time. They're actually not small. We will look at some in a few seconds. We'll try to get something that actually, represents a full model of the microbiome community for each sample. It's actually not that hard. So that's like normal Python code here. So basically we import from MICOM, we import the build workflow. We could also import community. We're not really going to use it, and pandas, but we don't need it here. So sorry about that. We could even kind of remove that here. And then what do we need to build the models? Well, obviously we need our taxonomy information. We need a model database, and that's the one we downloaded. we need a folder, and in that folder, it will basically save one full microbiota community metabolic model for each sample that you have. We have to specify the solver. In this case, we specify OSQP. Then we can specify a cutoff. So we can say if something is less than 1%, for instance, in the abundance, we kind of ignore it. The logic here is that if something is very, very, very low abundant, it cannot really produce huge amounts of metabolites. There's like certain thermodynamic levels, like a single bacteria can simply not produce like micromolar concentrations in the gut. It's really hard. It would need to kind of really go against physics. So we can kind of use that to exclude it and make the models a bit smaller if we want to. We will be fairly aggressive here. We would say that everything that's like less than 2.5% abundance, so that's like a fraction. So percent is like a factor of 100, so that's 2.5% here. We will remove those. So we kind of stay with only the most abundant thing. And that's mostly here to be a bit faster with all the simulations that we do. In general, we actually do 0.01% or even less than that. So we really only remove things that are really, really low abundant. And we use 2 threads. It will automatically parallelize everything across the models for you and show you a progress bar also and how that's going. And that will kind of do its course here. And while that is running, let's go back and actually understand what's happening. So what does Mycom actually do that makes it a bit easier to get flux distributions? So like I said, like usually we can kind of look for the maximum biomass, and often that's quite good because maximum biomass for a single bacteria is usually a single value. So that's great. But what does growth actually mean for community, right? If it's usually we talk about growth rate for single bacteria, does a microbiota, like a community, also have a growth rate? And it does. You could measure the OD of a bacterial community of different bacteria, for instance, in the labs, the same way you measured for a single bacteria. And that overall growth rate for the entire microbial community is usually expressed by a community growth rate, and I will call that mu C. Like mu is usually what's used for growth rates and C for community. And it's given basically like as a weighted sum of the individual growth rates, and it's weighted by the relative abundance. Because, for instance, if you have 2 strains in there and both grow with a growth rate of 1 per hour, and they're both in the same abundance, and on average, this entire thing community all still grows by the growth rate of 1 per hour. But if one was twice as fast and more abundant, it would kind of be weighted more, like it would contribute more to the overall biomass that's produced. So that's pretty straightforward. So we could think, okay, well, we extend it in a way like we did for a single genome scale metabolic model. We just now maximize the community growth rate, right? But well, there's a little problem here. The thing is, there's infinitely many solutions for individual growth rates that are actually consistent with that. And we can build up an example here. Let's assume we have a really simple example, almost stupid. Like we have 2 E. coli strains in here, exactly the same abundance, both 50% in the community. And we already know that maybe the maximum community growth rate is 1. It's exactly the example that I just made, for instance. So kind of both of those growth rates weighted by the relative abundance has to be 1. But there's a lot of possibilities, right? So it could be that this one grows with a rate of 2, then it would be 0.5 multiplied by a growth rate of 2. And the first one doesn't grow at all. Average would be 1, that works. The other, we can turn it around. That one grows with a growth rate of 2, also works. And actually, if we kind of plot both of those possible growth rates, everything that lies on that line is completely feasible. Like all of that is fine. That all of that gives us the maximum community growth rates. And that's like not good, because now we get like ambiguity, and that basically means that our flux comb becomes way bigger. Like it's actually harder to identify those individual growth rates now. Like since we have like all of them, there's infinitely many just on that line, right? So that's not what we want. Intuitively, we could say, okay, and that's what we started off when we developed Mycom. We kind of said, okay, if there's exactly like similar strains and then the same abundance, we know nothing else, we kind of would assume they grow on the same rate, right? It's the same organism. So we tried out like certain methods, what would give us that result? That was actually like the motivation. And we actually showed that what you can do is a method of regularization. The idea of regularization, we actually saw that today in the Parsimonious FBA. It means I take additional data or additional information about the biological system that will help me to reduce the solution to like a smaller subspace. And the information that we took in is that we said, well, like if you measure that a particular organism is present in your system, it has to grow. So it should not have a zero growth rate, but it's kind of intuitive, I would say. And we checked which regularization method would kind of work. And we proved that actually no linear method can ensure that it's actually true. If your abundance data shows that it is not, it like cannot be consistent with your abundance data, but a quadratic term actually does. So what you can do is you can minimize the sum of squares of the growth rates. And if you do so, it will try to it's like seems weird, why would we do that? But like an intuitive representation is it tries to actually distribute growth over as many organisms in the system as possible. For instance, for our example, it actually ends up with the point where it says, okay, both grow at the growth rate of 1, exactly the same, and the community growth rate is still 1. It reduces that infinite solution space and growth rate to a single point. And then we kind of also observed that you can also have the case like, basically, those bacteria still compete, right? So a single bacteria might not be too happy to actually sacrifice its own growth completely, basically to die off just to maximize the community growth rate. So often what you get is like some suboptimal community growth. You might not actually hit the maximum growth of the entire microbiome community. You hit something that's a little bit smaller, but ensures that everybody can grow because they're still competing for resources and want to survive. So that's basically, you can do that and you basically say, okay, I don't want to reach maybe the maximum growth rate, but another one. In that case, it kind of gives you the same thing. So if you say 80% optimum growth, you kind of get like a little bit slower growth rates. And that can kind of like really affect that. So that's basically what Mycom uses. But that's kind of, okay, I argumented here with a lot of like maybes and stuff. So you might actually ask if that works. And that method was actually called cooperative trade-off FBA. Cooperative because that regularization basically ensures that everybody can grow, which is kind of cooperative, and trade-off because you make a trade-off on the maximum growth rate. So that basically allows us to treat metagenome scale models with the same method as genome scale metabolic models because it gives us a unique growth rate for every single organism in the system. And that's really similar to normal FBA. So then I can use Parsimonious FBA and minimal media, all those cool methods that like FBA usually allow us without having to deal with that vastly increased like flux cone. But does it work? That's the main question. So the first thing is like I claimed that if we do that regularization, we get growth for more taxa. So that's the first thing that we looked at. So that's basically the density distribution of growth rates. And you see if we do nothing, if we just maximize community growth rate and take whatever solution the solver gives you, like what will happen is that the numerical solver will give you some solution that's easy for it to get, that definitely lies in that huge flux cone. But is it something that seems reasonable? Well, what we observed is that very few taxa, in most cases only a single taxa would grow at a really high growth rate. Everything else 10 to the minus 15, essentially absence of growth. It's not even there. So there was nothing. If we maintained the maximum growth rate and introduced just that regularization, we saw a massive shift already. Like you see now that suddenly a lot more taxa can actually grow. And if you actually like pull down the trade-off, it's really easy to get into a case where all of the observed taxa in the system can grow. And that is consistent with the 16S data. We observe those taxa, they live in the gut, they're able to grow, and they can grow, but you will not find those solutions if you just like don't do like the corporate trade-off. And then we also try to validate that. So since Mycom basically selects growth rates, like one of the obvious things was to see if those growth rates like kind of correspond to what we have. But how do you get growth rates for bacteria growing in the gut? Well, in that particular data set, we actually use metagenome samples where we get the entire genome. And bacterial genomes usually replicate from one origin of replication, and then the circular genome gets kind of replicated completely. So if you map the reads and you count how many reads map to a certain area on the genome, that's often called the coverage profile. It tells you like how much of that genome is covered at what rate at particular points. And what you usually see is that obviously since it starts replicating on the origin, you have like more reads there. and you kind of see that decay like to the end of the genome. So, and you can use that slope basically to estimate the replication rate. And that was a science paper from the Seagal lab. Maybe CTAs can post that in the channel. And they have actually shown in that paper that it actually does reproduce the actual growth rate. This is replication rate. It's not directly growth rate, but often those are kind of have to be aligned, right? If you like duplicate and you haven't duplicated your genome, you're in trouble, right? So and what we saw is like we kind of looked at what trade-off would actually give us the best agreement. And we saw that if we don't do any regularization and let's take like that random solution that we get from the solver, it actually doesn't reproduce growth rates really well. And then if we still assume maximum community growth, it doesn't really get that much better. But then like at the trade-off here starting at 0.7, we actually identified 0.5 was like the optimum point here. We actually do get like a positive correlation with the replication rates that we got from the metagenome data. So obviously it's not perfect. One thing is that here we kind of didn't have any diet data. So we didn't really know what metabolites are present in the gut to be eaten by bacteria. So we assumed like an average Western diet for every individual in that study. And that was obviously not true because people do eat differently. So we wouldn't have expected perfect agreement, but we would have expected some agreement. And we see that really big difference here of using that method over like just naively optimizing community growth rate. So it kind of worked. So easy peasy. So what's taking so long then usually? Well, metagenome scale models are really big. So that's kind of what, for instance, a realistic one looks like that we usually have. So what I kind of plotted here is that everything is a metabolite, and blue points here are reactions, and if they're connected, basically a metabolite gets consumed by a particular reaction. You can't even see the points because there's so many of them. In total, we have like 70,000 biochemical reactions going on. There's 47,000 metabolites. It's so many metabolites because we consider metabolite glucose inside of E. coli something different than glucose inside of Bacteroides, for instance. And there's like 300,000 connections between them. So it's pretty massive what has to be modeled. And you kind of see in that visualization, actually, the kind of really interconnected parts are actually the individual genera. For instance, here you kind of see imports from the outside, and then like the kind of empty space in the middle, pretty much, those are exchanges between microbes where they exchange metabolites. So that's like a huge problem to solve in mathematical terms. So if we go back to our notebook, it's actually done building it, and it will give us an output, which is a manifest. And that's really good to do some checks right now. So basically tells us, okay, for that sample in the database, I found 5 of the 5 taxa that you actually still had left in your model. Since we had that really aggressive cutoff, we're left with kind of not that many genera, and tells you also how much of the taxa we identified. And we see in the worst case, we still find 9 out of the 11 taxa in the database. There might be some that we can't find. So that's usually pretty good. And it also will calculate it in abundance because that's only like looking at the taxa. But for instance, a taxa that's really, really low abundant, you might be okay not being found in the database compared to like the most abundant taxon. So you kind of want to have that expressed also in abundance. And here it tells you that at least 80% usually of the overall abundance in our sample is also represented by the database. So that looks good. Like usually we say more than 50 is even okay. If it's less than 50, obviously you're not representing the actual biological system at all anymore. So that would be problematic. So to kind of start growth, like I said, we actually need to give information about which metabolites can be consumed, which is very different from other methods. And I think it's actually a plus here that you have to be explicit and say, okay, I assume there are starches actually that can be consumed. And what we will load in is the average Western diet. Since we know nothing about those individuals in their diet, we can kind of assume since they are on the Western continents that they might have eaten an average diet. So let's just assume that. We can load that in, it will download it. And that's actually what we will use as a growth medium. And let's look how that actually looks like. So we can, again, use load chime medium from the MICOM chime formats part. You just put in the file name and it will tell you what it looks like. So what we see now is we see a whole bunch of reactions here, which are the input, the fluxes for the export and import reactions. In this case, it only signifies imports. So flux is the actual rate by which something can be imported at its maximal rate. It's a bound. It's not the actual flux. So as we saw before, we have those like flux bounds on each flux that usually tell us if often those are just thermodynamic constraints. You make them really big. We just say, okay, like you can't be faster than diffusion, for instance. In this case, it's kind of based on the abundance of those things in an average diet, kind of calculated over 24 hours. We also have like a dilution thing. So what we did in the paper is actually, since most of the stuff gets absorbed in the small intestine, it doesn't actually get into the large intestine that easily. So we kind of depleted those as well. And we assumed that like 90% of that gets removed to the small intestine. And we actually saw the metabolites that are related to it. For instance, Fru-M is fructose and GLC-M is glucose. That's kind of the amounts that we have in here. It's really, we actually don't get a lot of glucose in the large intestine because it gets absorbed in the small intestine. So it's kind of adjusted for that. And with that diet, we actually start simulating growth. So we had the growth results. That's what we want to get. Our manifest is the output from before. It kind of just tells us what models and what samples we have. We have a model folder. That's exactly the same that we used before to save the models in. So that folder, and we can look at it. If we go to materials, we have that models folder. We see those weird pickle files, and that's literally what it is. It's like a canned model. So they're all in, like, and we have one for each sample and the manifest as well, if you want to look at that. So we have to specify a trade-off value, right? So if we go back to what I showed you in the validation, we did it actually with actual replication rates, and then we chose the trade-off that actually works out the best. But we did observe that you don't need to do that. Usually that one coincides pretty much with the one that allows growth for the most taxa. So if you wanted to choose that, you can just go through, test a few different trade-off values and use the one that allows growth for hopefully all taxa. But also we observed that 0.5 usually works pretty well. So we can kind of start with that. And it's the same, we can give it 2 threads because we have 2 CPUs. And we also save that using a Python library called Pickle, which will kind of compress the output and save it for later use in case you want to use it. Exactly. That's a cool Pickle GIF. So that will take about 3 minutes now. So yeah, that shouldn't be too long. In case you don't want to run that, you don't want to wait, you can also run the second one. So if you don't run that one, the next comment here will actually detect if that growth results have been generated. If they haven't generated, we'll just load like a cached version, but it's not taking too long. You usually also get like that indicator here. So it tells, oh, I took 30 seconds for the first model. I approximate that I will need more, maybe like 3 minutes more to go through all of them. So if you have more cores, more CPUs, that will run across as many as you want. So actually, since it kind of processes every single sample individually, that parallelizes really well, at least with many samples. So if you have 100 samples, you can run that on 100 cores if you want, and it will be really fast. Like it will be done maybe in a few minutes, even for large models. So it's really not that bad. You need some memory for that. So a good rule of thumb, if you want to run it on real models where you like shows a cutoff, like abundance cutoff that's actually lower, like 0.1%, is like 1 gigabyte per model. That works really well. So if you want to process 100 samples in parallel, then you will need about 100 gigabytes of RAM as well. But if you have a computer that has 100 CPUs, it probably has 100 gigabytes of RAM as well. That would be like already a massive computation server. But yeah, it does parallelize really well. It's just a bit slow here because we don't have that many CPUs. But I mean, we're halfway through just me babbling and probably will be done soon. But maybe let's already kind of see what we will get out of that. So that simulates growth. And what it actually does is it actually uses the same concept of parsimoniety, but it mostly uses it for imports. So in the gut, bacteria compete with our intestine for resources, right? We have a huge surface area of intestine in there that just absorbs metabolites like crazy if it can. So if you are a bacteria there and you want glucose, for instance, you have to be faster than that. So you have to be really quick in absorbing it. And that kind of means you want to use the smallest amount possible, because if you can grow on less glucose, then it's cheaper for you to get what you need to get maximum growth. So it's the same concept of parsimony. Like bacteria like to be efficient. Why should you import more glucose than you need for maximum growth? It doesn't really make sense. So it will do that as well for each model, and that will actually pinpoint the export fluxes well better than just not doing that. So what you will get is actually 3 sub-data sets in the growth results. One, you obviously get the predicted growth rate for all taxonoid samples. That often correlates a lot with abundance, so it's informative. Like it tells you if, for instance, if you have inconsistencies, if you see that something is predicted to not be able to grow even in low trade-off values, that often means that your assumption about the diet might be wrong. It also gives you import and export fluxes for each taxon and the external environment. So that's really interesting. It will tell you which part of the diet is actually preferentially taken up by that particular sample. Even if we have the same diet for each sample, one of them might, for instance, require more starch than another, and you will see that. And it will also give us annotations for fluxes. So that's quite cool. So let's look at that. In my case, it finished, and it should finish for you pretty soon as well, I hope. So let's look at the growth rates. How does that look? So we basically see the genus, we see the abundance, and then we also see the predicted growth rate. That unit of the growth rate is 1 per hour. We see also the number of metabolic reactions in that particular genus, the number of metabolites, the trade-offs that we used, and the sample ID. So we kind of know that Bacteroides in that sample grows at that rate. It's already cool, but I think like the most interesting part are the exchanges for me. So you can kind of see that, for instance, Faecalibacterium in that particular sample produce, for instance, that particular metabolite, and we will look at how to actually identify it at that rate. And basically, if it's positive, it usually means it gets exported or produced, and if it's negative, it means consumed, for instance. So you kind of know what it like consumes and produces, and like it's a very detailed view. You see that you have 31,000 exchange fluxes here, just that you get. This is not the internal fluxes that happen inside of the bacteria. It's just exchanges from a bacteria with its environment, and that's already like kind of very detailed view of the system. And it also locks the tolerance for you, so you kind of know when to ignore a certain flux because it's too small. But like, how could we actually tell what that is? Like, that's not really super informative here. Even if it tells me the metabolite is 1, 2D, well, I don't know what that is, right? So for instance, we might be interested in metabolite that's called AC. Well, what is that? Is that air conditioning maybe? That's quite some feature for a bacteria, but well, let's look it up. See, one thing that the growth results also contain is an annotation, and that's like quite helpful to map it also to external databases. So essentially that's the pandas data frame, and you can look up like this individual name, and it will tell you, okay, what is that? Well, the metabolite name, okay, we know that is AC. Oh, it's acetate. It's not air conditioning. Okay, makes sense. You also get like cross-links to certain databases. For instance, the human metabolite database. You actually get an INCHI screen. That's basically like a short representation of the chemical formula. You get a CAC compound number. If you want to look it up in CAC, you have a PubChem compound number, and you get the reaction that actually imports it. So that might be helpful. What you can also do is, if you want even more information, Agora has that really cool website, and I'll show you how that looks. Let's open that in a new tab, which is called the Virtual Metabolic Human, and Ines will probably talk about that tomorrow. So I'm not gonna take that away, but if you go and start, you can basically go here on home and say, Map Navigator, browse gut microbiota. And you can look for species, but you can also look for metabolites. So for instance, if I enter AC here, oops, maybe might have to scroll a little bit to find it. You can also use acetate here. You find the same thing. It says it's acetic acid, and you actually get even way more information. You get a full description, a full text, what it is. You get a lot of additional databases, and what's also kind of fun is you can actually look at which bacteria produce it. So you can, for instance, say present in which microbe reaction. That tells you every single biochemical reaction where that is used in a microbe. You can also say in which microbes it's present, and we see it's basically present in all of them. So Agora right now is 818 strains, and every single one knows acetate, so it's always there. You can also see where it's a fermentation product, and yeah, you can even look into human reactions that have it. So if you want to see, oh, how could that affect a human? Let's see, oh, okay, those are all the enzymes that humans have where it's actually acetate is involved. So you can get a lot of information about that back by that. And yeah, we have our growth data. So let's actually look what we got, right? The first thing we want to actually look at is the growth rates a little bit. So in Mycom, we can generate visualizations that are really similar to QIIME visualizations, only that we don't need to use the QIIME viewer. It's just like an HTML file, essentially a website. You can download and open that with a browser, or often it allows you to open it directly in your browser. So for instance, the first one is we can use the plot growth function from the Mycom viz sub package and kind of show the growth results. So if we do that, we actually get a file generated here, which looks like that. So you get growth rates and then the current date, and it ends in HTML. So it's a web page. You can download that. And if you have it, you might want to open it up with your browser if you can. And if you do that, it basically shows you what it describes. So you basically see each point is a single sample, and you see the growth rate of each of the genera in that sample. If you click on a single point, it will show you only the growth rate for that particular sample. So for instance, what we see here globally, and you can double click in an empty space to kind of reset it, a Bacteroides seems to be the fastest grower in that system. And it's followed actually by Fusobacterium. And Fusobacterium isn't really present that often, but it is a bit, it does grow fast in the sample where it's present. So that kind of means that it's expanding really quickly. And sometimes you see that separation as well. And you could, for instance, try to track which ones are healthy and which ones are sick. But yeah, basically it will rank like and show you which are the fastest and slowest growers in the system. That's okay. It's informative, but we might be more interested in what we can do with the exchange rates, right? So one thing is to look at growth niches. So the first thing we could do is just check every single sample. If you have questions, I think the Slack might be better if you have access to that, because I can't see the chat in Zoom right now when I'm presenting my slides. So go ahead and people will help you for sure. So the first thing we can try to ask is if we can actually just see like what is preferentially consumed in every single sample, and that's basically the plot exchanges per sample. So if we do that, same as before, we get like another HTML page in here, one that's called sample exchanges. And if you download that one, we open this one, it looks kind of like that. So it will basically show us for every single metabolite in that system. And if you hover over, it actually gives you the full name of the metabolite. Yeah. Oh, you can see. Oh, yeah, because the different window. I'm so sorry. Thanks for that. Let me try to rectify this. I will stop sharing my machine for a second and try to actually share my entire screen. This should be the one. Okay. Can you see it now? Yeah, perfect. Thanks so much. And thanks for raising your hand and bringing that up. I will actually, I think, yeah, you can just look at the growth rates yourself as well. So that kind of shows you the metabolite utilization. So we assumed the same diet for every sample, but that doesn't really mean as that much of the particular metabolite actually gets consumed. That's just an upper bound. We actually see, for instance, that here for AMP, adenosine monophosphate, we see it's like, for instance, only imported in a few of the samples, but not in others. And there's a few more of those. So you get the entire profile here and the samples on the bottom. They actually grouped like so that similar samples are next to each other. So we do see quite some heterogeneity here. So you do see, even if you give like different microbiomes the same diet, they will eat different things because it's different bacteria, they have different preferences. And what also is like really interesting is to look at what individual microbes consume, right? So that goes into really the idea of a growth niche. So basically we want to see is a certain bacteria that eat similar things as other bacteria. So do they overlap in their consumption of particular metabolites? And that can be done by the exchanges per taxon. What that does is actually, since it's really high dimensional, like what each bacteria eats is a lot of different fluxes, what it will do is it basically does a 2D plot with a similar idea as the embedding that we saw in the first tutorial. It's the same idea. Basically, it will arrange bacteria close to each other if they consume similar metabolites, and it will not do it if they consume very different metabolites. So those will be far together. And it uses a method called t-SNE that is really good at doing that, basically. So if we generate that, you get like another plot, and this one is now called taxon exchanges. So you can do the same thing, download this file, just open it up directly. Let me move this around. And this looks something like that. Like I said, it's like a projection. Essentially, if you're in the middle of that map, that means like you're kind of consuming similar things. And if you do that, you can also just click on one and you get only a single genus. So you kind of see that usually they kind of occupy similar growth niches. For instance, all the Bacteroides kind of are close to each other in each sample. But they might actually overlap with others like Lactobacillus here. You can double click again to set it back. And you do see there might be outliers. So there's one Coprococcus that kind of consumes very different things here. And this can be quite interesting, especially if you're looking for like a particular organism. You want to see if it, for instance, competes with a lot of different bacteria, or is it really its own thing, or does it adapt and kind of move its preference a little bit? So let's see if we can maybe find Clostridium here. One thing about Clostridium is that we see it does differentiate a little bit. And that's if that pathogen is present, like that might include C. diff. We actually don't know. Clostridium is a pretty large group, but we see it kind of does compete with a lot of different microbes here. So there's certain other microbes that are close to it. And that might be like a reason why it kind of prefers maybe, for instance, to be kind of more isolated, because that kind of eliminates competition. And it can definitely get higher growth values. And yeah, maybe the last thing we might be very interested in is metabolic connections to a phenotype. But maybe before I do that, I'll just show you what we found for that later data set. So basically, when we looked at the same plots that I showed you for like a large data set of like healthy and diabetic individuals, and we first looked at like the consumption preferences, we actually found that independent of whether the individuals were sick or not, there actually is like kind of almost a bimodality. Where kind of the microbiome either prefers to eat off more fibers and starches, or it prefers to eat more branched-chain amino acids and amino acids in general. So there was kind of that preference switch where a few samples seem to kind of like to eat more protein and some others like more fibers and starches. And then when we actually did like the same niche plot there, we actually saw that mostly everybody stays within their lane. Like all of the genera kind of occupy a very distinct point in the niche space, which kind of makes sense because we know that like the best thing you can do if you get into a new ecosystem is actually find an unoccupied niche. So you go into a space where you're not competing with anybody. But we did see that there's like definitely areas in the center where things at least get close. And there's some interesting ones that we saw here where like things that often like you don't see together kind of also seem to prefer similar growth niches. Okay. And yeah, when we looked at basically, we actually used the same thing. So when we looked at those export rates, for instance, and overall production of butyrate, the capacity, for instance, we saw that as it has been observed, like in real measurements, like we saw that short chain fatty acids, which are some compounds that are really important for the gut, they're produced by microbes and they usually help to differentiate our intestinal cells and also keeps them healthy. They have some differences, like for instance, we saw there was less butyrate production in type 1 diabetic individuals compared to controls. And we could actually also look at which bacteria produce those short chain fatty acids. One thing we observed is that for a lot of them, actually had quite a huge spread. So depending on the sample, actually a different genus might take it up It's like they're all kind of, for instance, produce more butyrate, but then like not the same genera necessarily do it. And that I think is like, yeah, that was important because like a lot of times, like when we do taxonomy analysis, we do things like saying, oh, Roseburia is a butyrate producer, so that's why it's beneficial. But actually the Roseburia might or might not produce butyrate depending on the diets that you give it and which other bacteria that's there. So that has to be taken into account here. And yeah, we can actually use that to try to find connections to a phenotype. So what we can do is we can look at the production rates, and I have that here, I think. So what we often look at is the overproduction flux. So there's two things we could look at. We could look at like the excess production. So basically there are metabolites that get exported into extracellular space in the gut, and then they get not taken up by other bacteria. And then there's like an excess of it, and you could look at that. But I think more important is like the over rate of a certain metabolite that it gets exported in the extracellular space, because even if it gets taken up by other bacteria, that is the pool that usually competes with intestinal cells. And one thing you might have noticed is that Mycom right now does not have a model for an intestinal cell in there. So there's no real competition for the microbes with intestine. So usually, and you can show that with the balance equations as well, but maybe you just believe me, that this is the pool that the intestine would actually have access to. So that's often really, we saw that it in general is more related to the phenotype. So that's like, for instance, the overall rate of butyrate that's produced by bacteria in the system. And that is actually what we can use as well to get a connection with the phenotype in the next part. And basically what it will ask you to do is, again, you need to pass in the phenotype that you're interested in. What we can do is we already wrote, like we already saved the manifest data and read it. So we can basically just, we will read it, we will set the index, that's not really important, but then we kind of choose again the disease stat column basically as our phenotype. So we just make a plot for that. We use the growth results, the phenotype, we define a tolerance, and we will use the 10 to the minus 3, and we say we want production fluxes. And if we do that, we'll actually fit a logistic regression. Don't worry about the warnings. That's all fine. Nothing burned here. And it will actually try to find fluxes that are predictive of the particular phenotype. But now it's not really bacteria. It will tell you exactly, okay, the microbiome community produces that particular metabolite, and that will give you will predict actually the phenotype. And if you run that, it will generate that fit file here, fit 2020, which is like the logistic regression model. You can open that up. And what does it give you? The first thing is it tells you only the coefficients that were actually predictive, basically coefficients that were above a certain threshold. And you could even download the data. That works for all visualizations actually that are generated. You can always download the raw data underlying each plot. And we can look what we get here. So we have law rate here, and that has a negative coefficient. A negative coefficient means associated with the first group. The first group is recurrent C. diff infection in that case, because it starts with a capital R. So that's like the, the alphabetic order that it uses. So it says like law rate here is associated with recurrent C. diff infection. Okay, we might have to look what that is. Pyruvate. Okay, that's like we know that this is something that gets produced in glycolysis, but also somehow related to growth rates. We see a little bit of aspartate, tetradecanoate, that's like a fatty acid, and maltotriose. So let's maybe in a second look what that might mean. And you can actually go and compare it for each of them. For instance, if you want to compare it for aspartate, it will show you the levels. That's law rates, the first one, and we can definitely see that this flux seems to be slightly higher, the overproduction flux in recurrent C. diff infection. And you can go through maltotriose as well, and you will find kind of the opposite, right? It's higher in healthy individuals. And then what it actually does is it will fit models, but will actually try to be a bit more conservative of what we have seen in the CHIME output, for instance. So it will tell us the same thing. The training accuracy is perfect. I can, just by the export fluxes, I can perfectly predict who is healthy and who is not. But then what it also does a leave one out cross-validation. So it basically trains the entire regression, but leaving out a single individual. And then it tries to predict that individual and will tell you how well it does. And we see that's actually it's lower. It's like 75% accuracy. And that's kind of what we would expect because we know that one looks pretty similar to the healthy ones, but it has a huge variation. So there's a few of them you can predict really well and some you cannot predict really well. And it actually shows you also like that confusion plot where you kind of see, okay, of the recurrent C. diff infection ones, how were they predicted in that leave one out cross-validation? You see that 3 out of the 4 were predicted of being actually sick as they are, whereas like 1 of them was predicted as healthy. And we know probably by now which one that was. And we actually see that for healthy, we also do that mistake at least once. So it tries to be conservative about having this. So that's interesting, but does that mean something? Do those metabolites mean something? Well, I put you some like fun facts here, basically. Maltotriose is a breakdown product of alpha amylase. Alpha amylase is a major enzyme that basically breaks down starches. So if the bacteria want to get access to glucose, they actually don't get glucose. Like we said, the small intestine gets rid of that, like it absorbs us for ourselves. We don't give that to bacteria. But all of the complex starches we can't digest. Those get into the large intestine. Alpha amylase breaks it down, and one of those breakdown things is maltotriose, which is kind of a sugar. Laurate, which we saw is actually super associated with, well, not super, but is associated with recurrent C. diff infection. That is interesting because it has been known as like a fatty acid that actually increases serum cholesterol more than any other fatty acids. If you eat a lot of laurate, you do increase serum cholesterol levels. And that already is like interesting here. And why is that interesting? Because I told you that C. diff is really good at detecting if other bacteria are present, but I never told you how it actually does that. It actually has a really cool mechanism how it does that. One of the things that bacteria actually break down or like to break down a lot is bile acids. Bile acids are really abundant. Like our gallbladder produces those, and we use that usually to dissolve fats so we can actually absorb them in the small intestine. But a lot of those primary bile acids, which are the ones that the gallbladder produces, they end up in the gut. And a lot of them are conjugated with like amino acids that our bacteria love to cleave off. So they deconjugate them and convert them to secondary bile acids. And basically one mechanism for C. diff to know that it can proliferate is it detects higher primary bile acid levels. Since it knows that if you have like a really intact microbiome community, it will usually assimilate most of the primary bile acids into secondary ones. So primary bile acid levels are low. Okay, so that's the first part of the story. Okay, so C. diff uses primary bile acid levels to detect if it can proliferate. But how do primary bile acids get produced? They get produced from cholesterol. That's like one of the major endpoints for cholesterol is primary bile acids. So if you would, in theory, increase the production of cholesterol and it would kind of end up in higher levels primary bile acids, that would actually create a feedback here, right? Like in communities that creates more cholesterol, creates more primary bile acids, that creates more proliferation of C. diff. It's a hypothesis, or we have really little data here, but it's kind of a cool story, I think. Also, like aspartate, like why do we have higher aspartate production recurrent C. diff? Well, C. diff toxins actually, which are really produced by C. diff in high abundances, are both aspartate proteases. So maybe there's some connection here, but yeah. It's very little data, but it's hypothetical. And that actually would be something very consistent to try out. So that's the thing. Like if I look at the abundance of microbes, I can generate a lot of hypotheses. But here you actually get kind of a story you can actually try out. So it says that there's some connection with starches. It tells you like laurate might be something. So that's something you can actually test, like, and then maybe see if you can use that. So it can lead to actionable stuff, but you definitely need to validate it experimentally to know if it works. Okay. Now again, it's your turn. We have a few exercises here. They should be fairly straightforward. And we think we're better in time now even. That's amazing. And basically, just to introduce that. So one thing where Mycom also really shines, I think, and which we haven't shown so much now, is that it can actually do you can actually run analysis on a single sample. You can really do personalized analysis. Since you actually have that working model, you can, for instance, turn some knobs. What happens if I change the diet? What happens to the growth rate of particular bacteria? What happens to exchange fluxes? That you can do for every single sample. And I kind of want to illustrate that. That's what we will do in the exercises. So there are a few parts that I did for you. We can look at the manifest again, just to see what models we generated. And if you remember, we had that weird individual, well, not weird, actually lucky individual, because, well, or unlucky, depends on your point of view. Like it was that individual here that had recurrent C. diff infections of ends in 48, but it looked more like a healthy individual. Might mean it's like that individual is getting better, might mean it has something that even overrides those positive microbiome effects. So we might be interested in looking at that. And I'll just show you how to run everything by hand, basically. So what we could do is we can use the load pickle model to kind of open the canned model and kind of load it up. We can also open up the medium. So we load the medium again, we load the pickle, we apply the medium. It's fairly simple. You just say, oh, the medium is equal to the medium flux. Now that particular growth medium is applied to that community. And then you can start simulating. So we can start that. That doesn't really take that long. It actually tells us also, we can look at the model, it will give us some information. And it will also tell us if particular exchange reactions are missing in the model. So for instance, that particular model doesn't have imports for that. And that can be true. It's not really, it's just for you to kind of, so that warning basically exists because we sometimes saw people using like growth media specific to a model database. And if you mix them up, you might get it that not a single one of them can be found. So it's just there for that. You can look at the overall number of metabolites and also tells you which bacteria are in there. So all of the information you need. You usually get 2 for each bacteria. One is like the extracellular space. One is the cytosol. In compartments. That's kind of like physical barriers that we assume are in the system, kind of, and you need transporters to get across them. And what I want you to do basically in the first one is to take that model with that applied diet. So it has that average Western diet as a single individual. So what you could, for instance, look at is in that individual person, what is like kind of the competitive landscape. And one way to get that is, well, it's a model, we can modify it as we want. So one thing we can do is we can just say, okay, I will knock out a particular taxon. For instance, I could knock out Akkermansia and see what that does to the growth rate of every other bacteria. So if it lowers the growth rate of another bacteria, that actually means there was a positive or trophic interaction. Akkermansia was providing something to the system that helped somebody else to grow. If it actually increases, that's usually commonly competition, right? If you remove that bacteria, another one can grow faster. So that means they probably competed for the same resources before. So you can look at that, and you can look at that in an individual sample. So it gives you a bit more information than just doing correlation analysis across samples, for instance. So that's what I want to see here. And you can actually go to the Mycom documentation for that. And hopefully it's informative enough to help you. Just please note that it has that fraction argument, which is actually the trade-off value. So we use the trade-off value of 0.5. So maybe adjust that to be more realistic. And yeah, you can use that and run that here. And would be cool if we could visualize that. I think I once saw somebody plot a heat map with Seaborn in a tutorial. I wish I could remember where. That would be really helpful here, I think. And then in the second example, I kind of just want you to look at fluxes a little bit. We will actually run cooperative trade-off by hand. So I put that here for you. That will take a little bit because it actually runs PFBA on all fluxes. So you now get also internal fluxes inside of each cell, which is even more of them. You can look at the fluxes, and I want you to first identify the primary acetate producer in the system. So which organism is producing most of the acetate. And I want you to also like choose any other metabolites that you really like what's your favorite metabolite. Check it out in BMH Live, see if it gets produced by anything or consumed. And also maybe try to find the metabolites that are produced at the highest level, or the fluxes basically that are the highest. Like is there certain biochemical reactions that are happening more than others? And I put in an addendum to actually show you how to choose the trade-off value with like another example. In our example here, just to take that away was not important. Any trade-off value would have worked. Since we have that little taxon there, it turns out all of them can always grow, but yeah. So for instance, if you run the corporate trade-off by hand, it kind of gives you that really detailed analysis as well. You get the community growth rate, get individual growth rates again, and it actually now gives you a Fluxes argument, and you can look at that and gives you that huge matrix, but basically has a lot of 4,000 unique Fluxes in each of the organisms, and you can just compare them and look at them. Yeah, and we'll see each other back here at 12:30, I think, and show you some, no, or 12:45, I don't know. Does anybody have the time plan? It should tell you in the 12:30. Oh, 2:30. Yeah, 12:30. I'm also in a different time zone apparently. So 2:30. Those should be fairly fast, so that would be fine, and I'll show you the solutions. And yeah, thanks so much. And yeah, keep the questions coming.